Have you ever thought about the hidden genetic knowledge that forms, well, the foundation of our entire food system?
The actual blueprint for survival.
Exactly. We're talking about the ultimate safety net that decides if a global crop thrives or just collapses in a single season.
That genetic resilience is everything. It's the insurance policy against, the unpredictable.
So, what's our mission today?
Today, our mission is to give you a shortcut to understanding the molecular toolkit that plant breeders and conservationists use every day. We're diving into some pretty dense academic material, lecture transcripts, research notes, all focused on pre-breeding, genetic diversity, and population structure.
And we're going to distill how all this diversity is measured, managed, and maybe most importantly, recaptured. All of it driven by molecular marker data.
Yep, the hard numbers.
Our goal here is to unpack why genetic variation isn't just some biological curiosity. It's the absolute first line of defense for a species.
Okay, so to start, we really have to define the basic units. What are we studying?
The populations themselves.
Right. So first you have intraspecific populations. These are just individuals of the same species, all sharing the same gene pool. Pretty straightforward.
And then the second category, interspecific. And this one's important because it includes individuals from different species that can actually still hybridize.
Exactly. They might not look alike, but they can still swap genetic information, which is a huge deal.
Which brings us to the core question. Why do we even need to assess diversity in the first place? Why should you, the listener, care about the size of a gene pool?
Because it's the foundational step for every single strategic decision. Think about a breeding program that's seeing low genetic gains. They're trying everything.
New fertilizers, different irrigation?
Right, and nothing's working. An assessment might show they have a really narrow genetic basis. They're just crossing individuals that are fundamentally too similar.
So they're mixing vanilla with slightly different vanilla and wondering why it's not chocolate.
Precisely. They've hit a genetic plateau, and that connects directly to the biggest risk factor.
The danger of uniformity.
Absolutely. Low variability exposes crops to catastrophic risk. And we have some terrifying historical examples. The sources point to the potato late blight in Ireland.
The 1840s.
The crop was clonally propagated. Every single potato was a genetic copy of the next. When the disease showed up,
there was no resistance anywhere. It just wiped everything out. A complete systemic failure.
And that's not just ancient history. We saw a similar thing in Brazil with cocoa production, devastated by disease because it just lacked the genetic breadth to fight back.
So by maintaining diversity, we're basically keeping a library of solutions, searching for novel traits, maybe for disease resistance or even biofortification.
Traits that were probably thrown out decades ago when the only goal was yield, yield, yield.
Okay, so if our modern super high-yield varieties have accidentally lost these vital defensive genes, how do we get them back? I assume that's where pre-breeding comes in.
That's exactly its mission. Pre-breeding has two main goals. First, identification. Locating desirable genes that are locked away in what we call unadapted germplasm.
So think wild relatives or ancient land races. They might look scraggly and not produce much.
But they carry incredible genetic gold. They might survive a drought that kills a modern crop.
So they're tough, but not commercially useful on their own.
Exactly. So the second goal is transfer. We take those wild traits and move them into intermediate materials that breeders can actually use in their modern breeding lines.
It sounds like a really long process.
It is a very long process. You have to evaluate the wild sources, hybridize them, stabilize the material. The sources say this often takes 7 to 8 years.
7 to 8 years. Just to create the starting material.
Just for the starting material. It really underscores why we can't afford to lose this diversity in the first place. It takes almost a decade just to re-engineer that lost safety net.
Wow. 7 years. It really highlights how crucial it is to understand that diversity from the get-go, and that means understanding how plants, well, how they mate.
That's the biological foundation for the whole strategy. We define mating systems based on the natural selfing rate.
And we can break them down into 3 simple categories.
Right. First, you have autogamous species. They self-pollinate more than 95% of the time. Think rice, common bean, soybean.
They're fine with high levels of homozygosity, lots of identical genes.
Then you have the opposite, allogamous species with less than 5% selfing, maize, carrots, sunflowers. If you force them to self-pollinate, they often crash.
Right, because of what's called genetic load.
Yep, all those nasty recessive genes start showing up.
Hmm.
So a hybrid breeding is the standard for them. And then you have the mixed systems like cotton or sorghum that are somewhere in between.
Okay, so once we know the mating system, we can ask if a population is genetically stable. And this brings us to the theoretical starting point. Hardy-Weinberg equilibrium, HW.
HWE is the perfect world that never really exists in nature. It's this ideal state where allele frequencies just don't change from one generation to the next.
It assumes random mating or panmixia and no migration, no mutation, no selection.
Basically, no evolution.
And since nature is messy, HWE is really just a baseline to measure how much a real population deviates, which is where, and this is where it gets really interesting, Wright's equilibrium comes in.
Exactly. This is for the non-panmictic populations, the ones that are actually inbreeding. And this introduces the most essential correction factor, the fixation index, or F.
Okay, let's break down what F means for the listener.
There are three basic outcomes.
So if F equals 0, the population basically matches Hardy-Weinberg. It's randomly mating.
No surprises.
Right. But if F is greater than 0, that is the smoking gun for inbreeding. It means you're seeing fewer heterozygotes, fewer mixed gene pairs than you'd expect.
And more homozygotes.
Exactly. And F of 1 would be extreme total selfing, no heterozygotes left at all.
And the rare case, F less than 0?
That means more heterozygotes than expected. It can happen, but usually we're focused on that F greater than 0 value. The fixation index is this incredibly practical tool. It tells you the genetic health of a population right now. And we saw a perfect case study of this in the sources. The piquia tree in the Brazilian Amazon.
A fantastic example. Researchers used codominant markers to study two different populations of this tree, and they used the fixation index, F and F, to calculate the apparent outcrossing rate.
So basically, how often are these trees successfully mating with a neighbor?
Precisely. And the comparison between the two sites was stark. The Topados population had an F value of 0.24.
Which translated to an outcrossing rate of about 61%.
Right. Now, compare that to the Rio Oro Prio population. Its F value was much lower, only 0.05.
Which is very close to 0, almost random mating.
And that gave it a high outcrossing rate of about 90%.
So what's the takeaway here?
The takeaway is that the Topados population is significantly more inbred. It's facing some kind of reproductive barrier, maybe habitat fragmentation, maybe fewer pollinators, and it's suffering from a sort of genetic hangover. That VEF value is a bright red flag for conservationists.
That one number tells them exactly which forest needs urgent help. Okay, so let's scale up. From individual trees to entire regions, we're moving into population structure.
Right. How is all that genetic variability organized, both within and between different populations?
And understanding that structure is critical for conservation.
It really is. Imagine the genome is a giant library. If all your rare genetic variants are like rare books stored on one massive shelf, your strategy is easy. But if they're scattered across 1,000 tiny shells all over the place,
you have to sample every single one to preserve the whole collection.
You got it. So structure analysis guides that strategy. We know that things like genetic drift and selection increase structure. They drive populations apart. Gene flow is the only thing that pulls them back together.
So how do we actually quantify these differences?
Well, we rely on metrics like Wright's F statistics and Nye's statistics. But for today, let's focus on two of the main measures you'll see.
Okay, let's start with FST.
So FST specifically measures the inbreeding that's due to the subdivision of populations. It asks, how different are the allele frequencies between subpopulation A and subpopulation B?
So if FST is 0?
If it's 0, they're basically one big happy family, a single shared gene pool. But as FST approaches 1...
What does that mean?
It means they're completely fixed for different alleles. Subpopulation A only has allele X, and subpopulation B only has allele Y. There is 0 gene flow. They are on separate evolutionary tracks.
That's the number that tells you if habitat fragmentation is causing permanent damage.
It is. Now, the second metric is Nye's GST, the coefficient of gene differentiation.
How is that different?
It's a similar idea, but it partitions the total gene diversity. It tells you what fraction of all the diversity you see is found within each subpopulation versus what fraction is found in the differences between them. And the really useful thing is that it comes with standardized benchmarks.
Oh, that's helpful. Let's give the listener that scale, because a number without context is just a number.
Okay, so if you calculate a GST value between 0 and 0.05, that's considered little or no genetic differentiation. But if you see a value greater than 0.20, that signifies very great genetic differentiation.
So a high number tells you that most of the action, most of the unique diversity, is found in the differences separating your groups.
Which reinforces that you need to sample broadly across all of those locations to capture it all.
Okay, that makes a ton of sense. Now let's zoom in even further, from the structure of groups to the relationships between individuals. We have to clarify how molecular difference is actually calculated because it really depends on the marker you use.
This is a crucial distinction. We have to separate dissimilarity from genetic distance. It all comes down to the kind of data you're working with.
So let's anchor this. Dissimilarity is like checking a box, right? Presence or absence.
Exactly. Dissimilarity is for binary data. A marker is present, a 1, or absent, a 0. This is typical of older dominant markers. You're just counting how many times one individual has a band that another one doesn't.
And genetic distance is more detailed. It's not just a checkbox.
Right. Genetic distance requires allele frequencies, so you need codominant markers, like microsatellites. These are the markers that can tell you if a plant is homozygous, has 2 copies of allele A, or heterozygous, with 1 copy of A and 1 of B.
You need that detail for the more complex formulas.
You do. So whether you calculate dissimilarity or genetic distance, you end up with this massive matrix of numbers showing how similar or different every individual is from every other one.
And what's the practical outcome of creating that huge matrix?
Clustering. You feed that matrix into an algorithm, and it produces a visual tree or a dendrogram. It's a diagram that visualizes the genetic relationships. And this is where the data either validates or challenges your assumptions about your samples.
And we saw a fantastic example of that in the sources with North American vaccinium species, blueberries.
A classic case. Researchers analyzed 66 individual blueberry plants from 3 different species using 14 codominant markers. They calculated the genetic distance, and then they clustered the results. The question was simple. Do the individuals group together according to their species label?
And what did the clustering reveal?
It revealed misclassifications. You'd have a plant that a botanist in the field labeled as species A, based on its leaves or flowers, but the molecular data showed it grouping strongly with species B.
So the DNA was telling a different story than the eyeballs were.
Exactly. It tells you the genetic definition of the species is much tighter than the visual one, or maybe that the 2 species are actively hybridizing out in the wild. This is the practical power, the genetic metrics questioning and confirming the true identity of your samples.
That really ties this whole deep dive together. The molecular toolkit gives you the final word.
It really is the highest fidelity data we have. We started with the absolute need for diversity and the 7-year slog of pre-breeding required to get it back.
Then we dove into the diagnostics, from the individual level with the fixation index F, all the way up to the landscape level with FST and GST, measuring how that variability is structured.
This whole toolkit is the shortcut to understanding the potential, the health, and the vulnerability of any plant population on Earth. It gives you the precise numbers you need to justify conservation and breeding.
And this leaves us with a final provocative thought. When we look at those high FST values, showing that strong differentiation between populations, what we're really looking at is an early warning system. These metrics are telling us if climate change and habitat fragmentation are pushing these subpopulations towards permanent, irreversible divergence. How much longer will gene flow be strong enough to keep these isolated patches from becoming entirely new and maybe genetically unviable species?
That is the urgent biological question those numbers are answering for us right now.